Okay, good morning, everyone. And again, a warm welcome to the campus of Prairie View A&M University. For those of you for whom it's the first time, welcome to campus. And some of you are returning, so very pleased to welcome you back. Again, my name is Gerard D'Souza. I have the pleasure and privilege of being the Dean of the College of Agriculture here at Prairie View A&M University. So on behalf of the College of Agriculture and Human Sciences, a warm welcome to the 26th Annual Dr. Raymond O. Berry Memorial Lecture and the 14th Annual Interdisciplinary Faculty of Reproductive Biology Retreat. Sounds very impressive. And I know you folks do a lot of great things. And I believe there's some 60 registrants in person, and some of the folks, I guess, are still working their way through the fog. I guarantee you it'll get better. As I promised some of you in the back, we're going to see if we can deliver a nice, warm, sunny day for you. I'll call upon my climate scientists in the college to fix it for us. Also want to welcome our virtual audience, including recently retired and now Director and Scientist Emeritus, Dr. Gary Newton. Gary, if you're there, in Kentucky, enjoying your retirement. Many of you know Dr. Newton. He's been around a long time, a few decades, and he's one of the reasons we are as prominent as we are at the International Goat Research Center and reproductive biology circles and more. And so, like I said, he's enjoying retirement in Kentucky, and I'm sure he is actively discovering new microbreweries. Those of you who know Dr. Newton. Warm welcome to you, Gary. Also, Dr. Foxworth, thanks for all you do in leading the Goat Center. Dr. Newton has passed the baton on to Dr. Foxworth, so he's continuing the great work. You know, and I do have to leave in a little bit because we have a simultaneous celebration or commemoration of World Food Day. I don't call it a celebration because amidst all the plenty and abundance, we have many places in Texas, the U.S., worldwide that are still hungry and food insecure. But I did want to say this Goat Research Center is a marvel. It is, I believe, the only comprehensive goat facility in the country that extends all the way from the reproductive biology and physiology and the genetics and the feed efficiency and the production protocols, all the way to value added through a newly renovated creamery that is nearing completion, but also a brand new meats lab on the farm that you're going to visit a little later today on your way to the Goat Center. And so, you know, we've got this comprehensive facility that really addresses the needs of our stakeholders. And in many ways, what you folks do in reproductive biology connects you to the stakeholders who need our assistance to overcome things like food security. So through our land-grant mission, both here on the campus of Prairie View as well as Texas A&M, we have land-grant universities, the only two in the state of Texas. So our mission extends beyond academics and beyond the classroom into the farms, into the fields, into the stomachs of people, and utilizing animals for a healthier planet and to make us all more food secure, more health secure, more climate secure, and much more. So believe it or not, you know, your discussion is very technical, and to an economist, really some of these terms don't always resonate, but I know what you folks are doing is important, and yet in many ways it's connected with real world and real life problems. So I hope that during the course of the day and beyond, you reflect on this strong connection between what you folks do in the labs and the needs of the stakeholders out there. And I know Dr. Foxworth gets this because in part I keep telling him we need to convert bucks into bucks. Okay, so I think there's a little bit of a joke there, but maybe you'll get it over the rest of the day. So in any case, I do want to welcome you again to campus to have an opportunity to tour our facilities and see some of the great things that are going on here on campus. And we're really growing in many ways. Clearly, our class of students here in the Ag College is not as strong as the College of Ag at Texas A&M. Indeed, our whole campus only has 9,000 students, and I believe Patrick Stover's college of Ag and Life Sciences, I think many of you are not in that college, but that's the college we work with most closely. They have as many students as we have at Prairie View, some 8,000 students, and still Patrick's not satisfied. That's kind of an aside, but we hope that we can increase the number of our students. So if you enjoy this experience, make sure you come back. Make sure you spread the word about some of the things going on here and some of the ways in which our students are collaborating. Indeed, we send our pre-vet students to Texas A&M to vet school, very successful from all indications. And we get some of your vet students here on campus to partner with us in terms of small ruminants and how to grow and expand that area. So once again, on behalf of the college, a warm welcome. And I hope to see you later today at the farm. I'm actually going to take Dr. Boast, who's the interim director of the Borlaug Institute on the Texas A&M campus. He's our keynote speaker at World Food Day, and he's never been on campus, so I'm going to take him around, and maybe we'll end up at the Goat Center, and you'll get an opportunity to visit with him briefly if you don't already know him. So thanks again, folks. Have a successful day, and see you soon.
Welcome you again. Good morning, everyone. So Dr. De Souza already acknowledged the welcome, but I really want to acknowledge everyone, welcome everyone to this 26th Dr. Raymond O'Barry Memorial Lecture and 14th IFRB retreat. So first, I really want to thank the peer review A&M University for providing this excellent auditorium and a lot of great support to make this meeting possible. My special thanks would like to give to the meeting organizers, Drs. Fuller Bazer, Greg Johnson, and Dr. Bill Foxworth. Thank you for putting up this outstanding program. And also I want to thank our trainees. Many of you came to help with setting up the posters this morning. Thank you. It's been great to see you all. So we are going to start with the IFRB retreat, which contains 2 faculty and 9 trainee presentations. So around 11:10 a.m., we're going to have a poster session in the International Goat Center. So lunch will be, I guess, will be served there. So we're going to come back after the lunch. Then we're going to continue with the retreat, which will conclude around 2:45 p.m. So I think this is a great opportunity. I feel really happy to see all of you and welcome again. And thank you for coming to this program. So I welcome Dr. Robert Berhardt to introduce our first speaker.
Good morning, everyone. It's my pleasure to begin this great sandwich of presentations today with the bread being Dr. Fuller Bazer at the beginning and Dr. Chinli Lee at the end. So our first presentation is going to be by Dr. Bazer, and the title of his presentation is The Multiple Roles of Arginine During Pregnancy. Dr. Bazer.
Okay, can everybody hear? Okay. So first of all, I'd like to thank the organizing committee for giving me a chance to give this talk today. This talk is going to be more of like a testimonial, and yeah. So this group that you see that you're looking at right now, we've been working together for about 25 years. And I think it's a testimonial to the IFRB and bringing people together that have common interests and common goals and have respect for each other's work and give credit for what each person contributes to the team. And today I did all the combinations of permutations between, if you put in PubMed between Burkhardt and Wu and Johnston and Bazer, you get 442 publications over the last 25 years, which is almost 18 papers per year. So not too bad. So for those of you who are young and getting out into the world, don't be afraid to collaborate. Work with others, and I think one of the great things about a team is that everybody's ego gets satisfied, and I think this is a way that we worked over the last 25 years, to some of us up to 29 years. Dr. Burkhardt and I started interacting when I first came here through being co-editors of the Biology of Reproduction, and have had good relationships, again, for the last 29, almost 30 years now. So arginine is the focus of this talk, and so it has a lot of different functions, and I wanted to go through all these, but just to indicate to you that the focus of a lot of the work that we've done over the last few years has focused on placental development and fetal development. And Dr. Wu is a nutritionist, of course, and we kind of sucked him into reproductive biology, and he's kind of moved us into nutrition. And so the combination of those two disciplines, I think, has been fairly productive, and that's what I'm going to focus on today. Unfortunately, last Friday, I gave a talk to Veterinary Physiology, Pharmacology, so some of you that were there will see a little repeat of what you're going to see today. So arginine is a precursor for nitric oxide, which is important for vasodilation and angiogenesis. Creatinine, we'll talk a little bit about. It's important for the urea cycle, so if you don't have a good urea cycle and you're consuming a lot of protein, you get hyperammonemia and die. And so this is very important. Can also be used to make proline, ornithine. Ornithine can be used to make polyamines. And then one of the things we discovered in the case of the sheep is that the sheep conceptus and uterus also produce agmatine that can be converted to polyamines. So arginine is important based on this work from Gwatkin in '66 and Martin and Sutherland in 2001, indicating that the mammalian embryo, as it goes from a morula stage embryo and begins to undergo compaction and forming a blastocyst, requires either arginine from morula to blastocyst stage. And so once this occurs, we have an intercell mass that's going to give rise to the embryo, of course, eventually to the chorion, or the outer layer of the membrane, and they will form the trophoblast, and extraembryonic endoderm will grow out into this area. So once this becomes polarized and the sodium-potassium HBases, for example, begin to function, then you can transport things like water and other nutrients into the blastocele, and the sodium gets pumped out to maintain these standing gradients that are necessary for this type of transport. Then, of course, in the large domestic animals that we work with, we go through this transition where after hatching from the zona pellucida, the embryo is going to go this process to reach a tubular form and then a filamentous form, and eventually will occupy a good part of the uterus. So during this period, there's a requirement for nutrients, secreted phosphoprotein 1, and these are involved in activating mTOR, or mechanistic target of rapamycin, which is associated with increasing cell proliferation and also mRNA translation to give you proteins that are important for this activity. So not only do these cells undergo proliferation, migration, but there's also obviously a lot of changes in the cytoskeletal elements that are associated with these nutrients, particularly arginine and secreted phosphoprotein 1 are involved in terms of stimulating what's called mTOR C2, which is associated with cytoskeletal elements. So over the years, we've focused on the interaction between interferon tau and progesterone, and today we'll just focus on this pathway here. because interferon tau is the pregnancy recognition signal in the case of the ruminant species, but a lot of people forget that it changes the phenotype of the uterus in the domestic animals because it silences all of the classical interferon-stimulated genes that are in the epithelial cells in close proximity to the conceptus, and upregulates a number of genes, for example, nutrient transporters, that are important for moving things like arginine and glucose into the uterine lumen for metabolism to support growth and development of the conceptus. And arginine being one of the more important ones of these that can stimulate mTOR, but also stimulate these other pathways that we have mentioned, and I'll talk more about those a little bit later. So this slide is just to indicate to you what I just got through saying, is that interferon tau, once it's produced, it will change the phenotype of the uterine epithelial cells so that these cells in blue are responsive to interferon tau, and they upregulate a transcription factor called interferon regulatory factor 2, which is a transcriptional repressor. So you no longer see any interferon-stimulated genes expressed by these epithelial cells that are in close proximity to the conceptus, whereas in the middle to deep glandular epithelial cells, RF2 is not expressed, and so you see all the classical interferon-stimulated genes being expressed in those cells in the mid to the deep uterine glandular epithelium. So they end up with three major classes of cells, if you like: the luminal and superficial glandular epithelial cells, the glandular epithelial cells, and the stromal cells, either the stratum compactum or the stratum spongiosum area of the uterus. And so all these enzymes, growth factors, cytokines, hormones, transport proteins, and nutrients, particularly today I'll talk about arginine and agmatine, become components of this intrauterine milieu that's going to be important for growth and development of the conceptus. So we showed a number of years ago by Hanjun Yao that if you look at the amount of arginine that you can recover from the uterine flushings of sheep during the estrous cycle, shown by these hatch bars, it doesn't really change during the course of the estrous cycle. So basically, the uterus is not getting one of the signals that it needs to transport. But in the case of pregnancy, where you have interferon tau coming into the picture, there's upregulation of transporters for arginine, and so you see an increase in arginine to day 15, and then as it gets metabolized, the levels begin to decrease, or did begin to decrease, in this study between day 15 and day 16. So this was important in terms of our thinking because, again, it took us to the beginning to think about arginine acting through the mTORC1, which is known to be very important for cell proliferation and migration and gene expression, also elongation and attachment of the conceptus in the case of these large domestic animals that is necessary for implantation and later for placentation. So arginine became a focus of the studies early on. So then Churchill, Jiaoqiu Wang, began to go through these and look at the SLC7A1 seemed to be the major transporter for getting arginine out of the uterine lumen into the trophectoderm. And then arginine could be then used to make nitric oxide, but nitric oxide synthase 3. And there's a cofactor for this called tetrahydrobiopterin. And so you really need both of these because tetrahydrobiopterin is a required cofactor for NOS3 to generate citrulline and also the nitric oxide. So we know that nitric oxide is important for implantation, angiogenesis, and embryonic growth and development from the literature. So if you knock out this system, then it's usually embryonic lethal. Similarly, arginine can go by arginase to ornithine, and ornithine decarboxylase converts ornithine into putrescine, which can then be metabolized to spermidine and spermine. So these are the polyamines. Again, if you knock out this pathway, in the case of animals, it's usually embryonic lethal. And some of the work done in mink, Bruce Murphy's group has shown, for example, if you don't get an increase in prolactin to stimulate growth and development of the blastocyst as it comes out of the delayed implantation, then the blastocysts don't develop. So we did some experiments which led us to discover this pathway for producing agmatine in the sheep. Because at the time we were doing this, agmatine had been shown to be produced in the brain of humans, for example, and also in the kidney, but nowhere else. And there weren't any reports of any agmatine production by the uterus or conceptus tissue of any domestic animals. And so the first study we did was to use a morpholino antisense oligonucleotide, or MAO. And so this basically blocks translation of the messenger RNA to give you ornithine decarboxylase. So we introduced the MAO into the uterus on day 8, and then we recovered the conceptuses on day 16. So we wanted to find out, and our hypothesis was that if we did this, that these conceptuses might reach the tubular stage but would not be able to go through and form a long filamentous conceptus. What we found is that we had the controls look normal, and we had one group we called, later we called MAO ODC1 knockout type A and then a type B. But if you look at the protein, so you can see the ornithine decarboxylase in the green here. So it is knocked down in both of these groups, and we also see a decrease in the amount of ODC on a per cell basis. And if you do immunohistochemistry, you also see that ornithine decarboxylase is less, much less than in the controls, and similar to what it is in the IgG control. So when we looked at this more carefully, again, this is Churchill's work. We showed that in the animals, in the MAO control, we had ADC mRNA was relatively low compared to what happened when you knock down ODC1. This seemed to upregulate the expression of the RNA for ornithine decarboxylase in both of these phenotypes. But the interesting thing was that of the significant change was that the agglutinin protein itself, agglutinase protein itself, was upregulated more in the A-type group that did elongate, similar to those in the control. So this suggested that this alternative pathway was able to rescue some of the conceptuses if it was upregulated sufficiently. And then we also looked at the fact that there's agglutinin in the uterine lumen of these sheep was significantly less than the B-type, and also if you look at the amount of agglutinin in the uterine flushings of these B-type embryos, that was also significantly reduced. So these results showed us that in addition to the classical ornithine decarboxylase pathway, that in the case of sheep, if we knock down ODC1 mRNA translation, that the sheep conceptus can upregulate the arginine decarboxylase pathway to produce agglutinin that can be used then to make polyamines. So then we went back and just take a minute on this slide to indicate that these are just normally pregnant sheep. So if you look between day 12 and day 16, you can see that polyamines are present. And if you look at agglutinin, it's also normally present. So it's not that the sheep conceptus can't normally make agglutinin, but it's just that it seems to be upregulated particularly well if the ornithine decarboxylase pathway is compromised. So the next step was to see, since this alternative pathway gave us a variable phenotype, whether if we knock down ODC1, ADC, and also agglutinase, if we knock down all three of these, what would be the results? And so in this study, we had the introduced morpholino antisense oligonucleotides for ornithine decarboxylase, arginine decarboxylase, and then both of these mRNAs on date, and then we looked on day 16. So again, covering this period of transition from a spherical to tubular to filamentous conceptus. So interesting, the MAOs in these species is only taken up by the trophectoderm. So the uterine epithelial cells do not take it up because they have such a low mitotic index, they're just not involved in taking up these this MAO. So again, what we found was MAO control was the normal. MAO, if you just knock down ODC1, again, we got a lot of the ones that were normal. If you just knock down arginine decarboxylase, most of them are somewhat normal. If you knock down both of them, the conceptuses tend to be more fragmented, but still they had at least initiated a long elongation process. But again, we had this B-type phenotype where if you look at the interferon production, which was our production output, if you like, it was significantly reduced only in the group where we knocked down all three of these and got this B-type phenotype. So why some of these still make it, but about 25% of the embryos in this particular study went to this stage, and the other 75% were knocked down. So if we think about another pathway that's important is the ornithine polyamine pathway, or the agglutinin to polyamine pathway that we just talked about. And so polyamines are very important, and just kind of started off with Catherine Halloran and Emily Hoskins' work, and then Claire Stenhouse is now involved in this, looking at the polyamines. But polyamines are important because of the fact that they're very important in mRNA stabilization, DNA stabilization, a lot of signal transduction, of course, required for cell growth and proliferation, and many other functions that you can see listed on this slide. So again, this is why if you knock out the polyamine pathway, since it's involved in all of these different pathways, then it's going to be compromised. So in finishing up some studies that Kitty and Claire have just got through doing, if you look at the interaction again between progesterone and interferon tau, we find in this study that progesterone seems to be involved primarily in upregulating the transporters for agmatine, putrescine, spermidine, and spermine. Whereas interferon tau is upregulating the ability of methionine to be used to make polyamines and different steps. And also, if you look on this side where there's an interconversion of polyamines from putrescine to spermidine, interferon tau is also upregulating the genes that are associated with this interconversion. So depending on the tissue you're looking at and the studies that you look at, either spermidine or spermine seems to be obviously the most important of these, and spermine oftentimes is considered to be the more active of these three polyamines, but it's also the least stable of these three polyamines. So anyway, this is a graphical abstract of a paper we just submitted to Biology of Reproduction, so we'll see how that goes. So the other thing is with arginine is, The other thing with arginine is the ability of arginine to produce creatine. So normally creatine is involved in this pathway where you get the production of ATP, and creatine is associated with cellular proliferation, cell migration, cytoskeletal remodeling, and in the embryo fetus it's associated with the biogenesis, that is the muscle development and also the neurological development in individuals. So you'll hear towards the end of the day today from Nerve. And so I just wanted to mention at this point that if you look at the adult, the kidney is involved in taking arginine and glycine and making what's called guanidino acetate. And guanidino acetate is then transported to the liver, and the liver converts this to creatine, and then creatine can go to target tissues like muscle and so forth. So all of these processes that we talk about are ATP dependent, and so it's not surprising that there's a system. But what's surprising is that whereas in adults you have the kidney and the liver having to participate to make creatine, in the case of the reproductive tract, pretty much all the components of making creatine are located within the pregnant uterus. So we have the creatine coming from the mother that can be transported across to the trophectoderm. We have arginine glycine that can be converted by the uterine endometrium to make guanidino acetate that can be transported across to the trophectoderm, and then the guanidino acetate here can be converted then to creatine. So again, Nerve will talk more about this this afternoon. So this slide just indicates the fact that arginine, as well as polyamines, can stimulate different cell signaling pathways. So we talked about the mTOR pathway that's associated with mRNA translation and ribosome biogenesis and cell proliferation. Also, the mTORC2 pathway is associated with cytoskeletal reorganization. So not only arginine, but also its product through the polyamine pathway can stimulate this, at least this mTORC1 pathway, and arginine plus secreted phosphoprotein 1 that Dr. Johnson's done a lot of work with interact to also act through this AKT pathway, and there's evidence that we published indicating the P38 pathway. P38 is a transcription factor that could also change the cell structure in terms of its ability to undergo migration and adhesion. And so this study is a study we've done with Dr. Wu with pigs, where you can feed arginine to sows, and you can increase the litter size, and you can increase the weight of the litter. And when we first started doing these studies, I was the one doing all the dissections. And so when you first go through this and have a little experience, you can actually start to pick out the treatment that the animals are on, because you can see that this is a control that has a fairly well-developed allantoic membrane, but the ones that are getting 0.4% L-arginine in their diet, much more proliferation of the allantoids. So keep in mind, in the chorioallantoids, all the blood vessels are in the allantoids. But in the 0.8% L-arginine, there was also an increase in vascularity, but not to the same extent as the 4%. So one of the recent breakthroughs from Dr. Wu's lab is to show that citrulline and glutamine are not metabolized in the rumen. And so this means that now we can feed to ruminant citrulline that when it goes into the body will get converted to arginine almost immediately, and hopefully give us some of the similar effects that we've seen in terms of the work that's been done with not only pigs, but also rodents and also humans. We're supplementing, for example, women with small for date babies with arginine, you can increase the birth weight of the offspring as much as 20%. And so this is the question we have now is whether or not dietary supplementation with creatine, with a citrulline, will result in increases in arginine and conceptus development in the case of the ruminant species. So again, I'd like to thank my people that I've been able to work with, and just indicate too, this is sort of a testimonial to teamwork, and I appreciate the opportunity to make this presentation. Thank you.
Thanks, Dr. Bazer. Our next presentation, as we begin this trainee presentations, is by Xin Fang, and she's a PhD student with Dr. Jingli Li. The title of her talk is Activation of TGF-beta signaling as an oncogenic switch in Sertoli cells in vivo.
Okay, thank you for the introduction. So today my presentation is about the activation of the TGF-beta signaling on the oncogenic switch in the Sertoli cells in vivo. So our lab is focused on the TGF-beta signaling pathways. The TGF-beta signaling can regulate a variety of the cellular properties, including but not limited to the cell growth, cell differentiation, cell migration, and cell apoptosis. In the TGF-beta signaling, the ligand will bind to the the ligand will bind to the receptors on the cell membranes and then phosphorylate the receptor responsive SMAD. For the TGF signaling will be SMAD 2 and 3, and for the BMP signaling pathway will be the SMAD 158. The phosphorylated SMAD will form a complex with the co-SMAD, which will be the SMAD 4, and translocate to the nucleus and regulate gene expression. So in the previous studies, our lab generated a mouse model with a constitutively activated TGF-beta receptor activation will be the sterile. So we did the microscopic and histological image of the tested from the mice bearing the activation of TGF-beta receptor 1. Here's a 2-month mouse tested. As you can see, there are several hemorrhagic spots in the testes, and in the 4 months, the testis size will be increased. And then we did HE staining in the testes. We found that the several tumor foci, they are surrounded with disrupted seminiferous tubules. And inside the tumor foci, they are full of the poorly differentiated granulosa-like cells. Because those mice are infertile, so we further look at the epididymis. As you can see in the controls, the sperm was stored in the lumen of epididymis, but no sperms were observed in the mice bearing the activation of the TGF-beta receptor 1. Thus, we know that the activation of the TGF-beta receptor 1 will cause the sex core stromal tumor formation and the defects in spermatogenesis. since the histological features of the testicular tumor resembles to the ovarian granulosa cell tumors. So we tested the expression of several granulosa cell markers, including the inhibin alpha, FOXL1, and FOXL2. Among these three markers, the FOXL2 is a lineage marker of granulosa cells. As you can see, the cells inside the tumor foci are intensively express these three markers. To further identify the tumor cell types, we also did the staining testing the SOX9, which is the Sertoli cell marker, and the CYP17A1, a Leydig cell markers. But the tumor cells inside the tumor foci were negatively stained with these two markers, which help us exclude the possibility of Sertoli cell tumor and Leydig cell tumor. Thus, by doing this staining, we know that the activation of TGF-beta receptor 1 induces the formation of granulosa cell tumor in the testes. A lot of studies have proved that the key signaling pathway and gene expression could induce the formation of testicular granulosa cells tumor, including the activation of the beta-catenin-induced FOXL2 expression, the PTEN loss induced AKT phosphorylation and the FOXO1A expression, and the loss of inhibin alpha can induce more SMAD3 expression and FSH signaling. Also, we know that Sertoli cell is critical for the spermatogenesis in the testes and involved in the granulosa cell tumor formation in the testes. Thus, we isolate the Sertoli cells from the mouse testes. To make sure we can successfully isolate the Sertoli cells, we did a staining testing the 3-beta-HSD, which is the Leydig cell markers, and the SOX9, a Sertoli cell marker. By doing this staining, we know that 90% of the isolated cells are the Sertoli cells. And then we did the qPCR using these cells and found the TGF-beta receptor 1, SOX9, they are changed in those cells. Also, the gene related to the Sertoli cell maturation, like the RHOX5 or mature Sertoli cell marker and NGFR or immature Sertoli cell marker, are significantly changed. And the SOX4, which is related to the cancer development, also changed in those Sertoli cells. So to further look at the molecular change in the Sertoli cells, we did RNA-seq using those isolated Sertoli cells. And we identified 3,700 differentially expressed genes in our RNA-seq data. And then we using the IPA to analyze the signaling pathway, found several pathways that relate to the testis functions are significantly changed, including the IL-6 signaling, FGF signaling, CXCR4, and the GDNF family ligand receptor interaction. They are all changed. And also the signaling pathway related to the cancer development also changed, including the NFKB, STAT3, ALK signaling pathway, and so on. So we did the data mining in our RNA-seq data and found several genes that is the altered TGF-beta signaling affect the gene expression that can change the Sertoli cell functions and development, including the blood testis barrier development and maintenance, cancer development, regulation of pluripotency of stem cells and spermatogenesis, and hormone signaling in the Sertoli cells. Also, the maturation of Sertoli are also severely affected by the activation of the TGF-beta signaling. So we come up a question, is the testicular granulosa cell tumor initiated from Sertoli cells? Then, since the first mouse model was driven by the AMHR2-CRE, which is expressed in the Sertoli cell and Leydig cells in the testes, we generate our second mouse model, which we using the AMH-CRE, which is specifically expressed in the Sertoli cells. And to be brief, I will call this mouse model as a CA mouse. And we confirmed the activation of the TGF-beta receptor 1 by doing the qPCR and the Western blot. Also, we tested the SMAD2 and SMAD3 phosphorylation by doing Western blot. We found in the CA mice, the SMAD2 and SMAD3 was significantly increased in those mice, suggesting we successfully generated this mouse model. And then we collect this mouse at the age of 2 months and 4 months. As you can see, it's very similar with the first mouse model. There are several hemorrhagic spots in the 2 months testes, and at 4 months, the size of testes significantly increased. Then we want to know that the activation of TGF-beta receptor 1 in the Sertoli cell alone can still initiate the granulocyte cell tumor or not. So we did a staining testing the granulocyte cell markers, the FOXL1 and FOXL2. As you can see in the tumor foci, they are intensely expressing the granulocyte cell markers and will not express the CYP17A1, which is the Leydig cell tumors. So Leydig cell markers. So we know that although we only activate TGF-beta receptor 1 in the Sertoli cell, it still initiates the granulocyte cell tumor in the testes. Then we also test whether there are still defects in spermatogenesis. So we test the marker as MSY2, which is a marker for the spermatogenesis. And we found very little cells were expressing the MSY2. Also the SOX9, the Sertoli cell markers. As you can see, the number of SOX9 positive cells are significantly increased. And also the CYP17A1 in the Leydig cells. Then we look at the distribution of some critical cell markers, the DMRT1 and FOXL2. The DMRT1 is a critical gene that can maintain the testis identity. In the previous studies, the loss of the DMRT1 can induce the expression of the FOXL2 and other female genes, which will induce the granulocyte cell tumor developed in the testis. The figure F and figure G are the serial sections. As you can see, the DMRT1 is in the control. The DMRT1 is expressed in the Sertoli cells and some of the spermatogonium. And in the CMIs, you can see the increasing number of the DMRT1 positive cells. But in this seminiferous tubule, you can see part of the cells losing the expression of the DMRT1. And in the serial sections, in these seminiferous tubules, there are a lot of cells start to express the FOXL2. So this may suggest that with the activation of the TGF-beta signaling, the Sertoli cells in the CMIs lose the expression of DMRT1 and could be reprogramming to a granulocyte cell, and it start to express the FOXL2. Also, we also did the staining of the SOX9, and the CYP17A1 also shows some changes in the Sertoli cell numbers and Leydig cells. Then we come out to next step. We generate the third mouse models, which can genetically label the CMIs to trace the testis granulocyte cell tumor origin. We cross the mice bearing the constitutively activated TGF-beta receptor mutant gene with the MTMG mouse model, together with the AMHR-CRE, which is the AMH-CRE, which is the Sertoli cell specific CRE. The MTMG mouse is the contains dual fluorescence. So in the control mice without CRE expression, the normal cells will express the MD tomato, which will be the red color. And with the CRE expressed in the Sertoli cells, these Sertoli cells will express the EGFP, which will be the green color. So we hope this mouse model could help us identify whether those tumors, based on the color, we can identify the tumors are initiated from the Sertoli cell or not. Then we collect the testis at the age of 4 months. As you can, and we want to confirm whether we successfully generate this mouse. So we did the staining in the D40 mouse testis, testing the EGFP. As you can see in the control mice without CRE expression, there's no signal for the GFP. But in the CMIs, the cells inside seminiferous tubule were expressing the GFP, suggesting the mouse model was kind of successful, and the SOX9 expression still observed in the Sertoli cells. So in the future, we are going to study the role of the TGF-beta signaling using the Sertoli cell line, such as the TM4 And also, we are going to use this CA with MTMG mouse model and sort the cells using the EGFP from this mouse and study the gene expression at protein and transcript level. I want to thank my mentor, Dr. Chunli Li, for his great guidance in my studies, and also Dr. Baylis, Dr. Rejenko, Dr. Ivanov, and also want to thank my lab members helping me complete all this work. Thank you so much. I'd like to take any questions. Hi. Thank you. The fibrotic changes, because so actually I think in the granulosa cell tumors I didn't saw a lot of fibrotic changes because most of them become the granulosa like cells. But I think that's a great idea. I will double check in the future. Yeah. Thank you.
Okay. Our next speaker is another trainee presentation, Dr. Gabby Delmaso, a Ph.D. student with Dr. Poehler. Title of her talk is Influence of Pregnancy-Associated Glycoproteins on the Reproductive Transcriptome in Boss and Indicus heifers.
All right. Good morning, everybody. Thank you for the introduction, Dr. Burkhardt. So it is very well established in cattle that by fertilization, we are expecting greater pregnancy rates compared to later stages of pregnancy. And of course, this is happening due to the occurrence of fetal losses and embryo losses throughout gestation. So embryonic losses can be divided into periods. We can have early embryonic mortality happening between days 8 to 30, and then late embryonic mortality happening between days 30 to 100 of gestation. So late embryonic mortality can be easily diagnosed because this is the period in which we have most of the tools available for pregnancy diagnosis. But even still, even with so many information available, we still don't study this period as much as we do compared to early embryonic mortality, and we believe this is the reason. So when we compare the rates, of course, we are expecting greater rates of early embryonic losses in beef and dairy herds. And of course, this is a very important period for pregnancy establishment and pregnancy maintenance. But late embryonic losses are also important because they will cost more to the producer, especially if we consider beef herds in which most of the income is derived from the calves that will be sold. So late embryonic mortality can have multiple causes, and we don't have time to go through all of them today, but I do want to point out that some of these causes are manageable. For example, if we consider causes related to estrous synchronization and inducing ovulation from small follicles, or even environmental factors related to metabolic stressors, nutrition, and infectious diseases. So again, we can manage that. We have data available showing that some sires have greater rates of embryonic mortality compared to others, and our lab is actually already working on the paternal contribution to pregnancy, which leave us today with inadequate placenta function. So we believe that late embryonic mortality is mainly caused by inadequate placenta function because this is happening during the period of active placentation. So if there's something wrong with the placenta, the embryo is going to be the first one to be compromised. So in cattle, between days 20 to 22, we will have the formation of these trophoblast giant cells in the trophectoderm portion, and these cells will start migrating into the luminal epithelium of the endometrium and occupying this endometrium, this epithelium, contributing to the formation of the placenta. So by the end of this migration process, these cells will also start releasing their contents into the maternal circulation. And between these contents, we can find molecules such as placental lactogen and also pregnancy-associated glycoproteins, also known as PEGs. So PEGs have been used for quite a while now to diagnose pregnancy, and they have also been recently pointed as potential tools to verify the placenta viability. So in 2013, Dr. Poehler reported in beef herds that pregnant cows on day 28 of gestation that had greater concentrations of PEGs would also have a greater probability of maintaining pregnancy compared with cows with lower PEG concentrations. And he also reported the same similar data in dairy herds. So if you look to this graph here, pregnant cows at day 31 that had greater PEG concentrations would have a lower probability of late embryonic mortality. A former student from our lab, Sydney Rees, also reported different PEG concentrations between cows that maintained pregnancy and cows that lost pregnancy. So as you can see, we had greater PEG concentrations in cows that maintained pregnancy. And she also reported different prostaglandin E profiles in which cows that maintained pregnancy had lower concentrations of PGE compared to cows that lost pregnancy between days 30 to 38. And this other group here in 2019 cultured endometrium explants with PEGs either for 24 or 96 hours, and they did report transcripts related to prostaglandin synthesis, matrix remodeling, and also some chemokines. So we're seeing here with these findings that PEGs are eliciting changes in the reproductive environment, but we don't have the details of these changes, when do they start, and how would they behave comparing environments with high PEG or low PEG concentrations. So in my project, my objective was to compare the molecular footprint of these concepts derived from high PEG pregnancies versus low PEG pregnancies. And we hypothesized that placentation will be compromised in these concepts derived from low PEG pregnancies, and also that all those markers for matrix and tissue remodeling, angiogenesis, vasculogenesis, and also those trophectoderm giant cell markers, or basically PEGs, would be downregulated in the low PEG group. So the design was pretty simple. We started here with a set of heifers on day 0, and they were all artificially inseminated. On day 25, half of these litters were slaughtered, and then the other half was slaughtered on day 36, and we collected blood samples from all of them to measure PEG concentrations. So we measure PEG concentrations using validated in-house ELISA from our lab. So with this PEG concentration, we were able to separate these heifers in groups of high and low PEGs, and of course, we only kept the pregnant heifers in this study. So on day 25, we were able to get 4 high PEG and 4 low PEG heifers, and on day 36, 5 high PEG and 3 low PEG heifers. So here are just graphs showing the progesterone concentrations and PEG concentrations in both days, and as you can see, we did not have any significant difference on progesterone concentrations, but we did have significant differences here in PEG concentrations for both days. So again, we were able to separate them in 2 groups, high PEG and low PEG. On the slaughter day, we collected reproductive tissues, so we were able to collect caruncles, trophectoderm, or cod ligaden according to the stage of pregnancy, and also the corpus luteum. So those samples were stored in RNA later. After removing RNA later, we just proceeded with the RNA extraction using a commercial kit. We verified RNA integrity and quality using the bioanalyzer, and then we submitted the samples for RNA sequencing. All the reads were aligned to the Bostaros genome. We performed all bioinformatics using HR, and then the gene ontology analysis using IPA and DAPIT. So here are some volcano plots just showing the differently expressed genes, the DEGs, on day 25. I'm showing here data from trophectoderm, caruncle, and CL. So everything that you see in blue today will be upregulated in the low PEG group compared to high, and everything in pink will be downregulated in the low PEG group compared to high. So as you could see, we did have some DEGs here in the trophectoderm and in the caruncle, but not a lot on the CL. So today, we'll be focusing more in these 2 tissues. So when we started looking to the overall processes that were up or downregulated in the low PEG group here in the trophectoderm, we did not see a lot of downregulated processes, but more upregulated processes, and most of them were related to embryonic development, organismal survival, and development. And when looking to the overall graphic representation of these major biological themes that were being represented, we did find some processes being inhibited. So in this graph here, everything that you see in blue is being inhibited. And we could see inhibited processes related to morbidity and mortality, neonatal, perinatal, organismal death, and also hypoplasia. So it seems that in the trophectoderm on day 25, there's nothing indicating to us that the embryo is being compromised. But when we go to the caruncule, although we could see some upregulated processes related to tissue and organism development, we also saw some processes downregulated related to embryonic development, immune cell immune response, and also the formation of the hematological system that was being downregulated in the low PEC group. And again, when looking to the overall representation of this, we did find these inhibited processes related to vasculature, vasculogenesis, and chemotaxis, and one enriched process related to perinatal death. So it seems that the caruncule is telling us a different story. After looking to the overall processes, we're starting looking for specific genes to try to prove our hypothesis. So we were looking for genes related to those extracellular matrix organization and angiogenesis, vasculogenesis, and PEG markers. So we did find an upregulation of those extracellular matrix organization genes in the trophectodermal D5, and also genes related to embryonic morphogenesis and development. And we also found just a few genes related to interferon response that were being downregulated. In the caruncule, we found genes upregulated related to the membrane components and downregulated related to the formation of the hematological system. So everything here agrees with those overall processes that we just found. And with regards to the trophoblast giant cell markers, so the PEGs, they were not being differently expressed between groups of high and low PEGs. But when I looked to their overall fold change, they were negative. So we can say that they were being lower expressed in the low PEG group. So when we go to the day 36, things starts getting more exciting. We did see more DEGs in the cotyledon and in the caruncule, but again, not a lot on the CL. So one more time, looking to the overall processes, we had, again, more downregulated processes too. And this time, the downregulated processes were related to organism development, tissue morphology, and organism survival. And when looking to the top 4 pathways, either up or downregulated, we thought it was really interesting to see the hyposignaling pathway being downregulated here on day 36. Usually this is related to early embryonic development, very well established in mice. So we are still investigating why this pathway would be downregulated here at day 36. When we go to the caruncule, not a lot of upregulated processes, but more downregulated processes in the low PEG group related to organ morphology, tissue morphology, and also organismal development and survival. And among those pathways that were being downregulated in the low PEG group, most of them were related to immune response and also a ferroptosis signaling pathway. So again, looking for the specific genes, we did find those extracellular matrix remodeling genes being upregulated again in the cotyledon, but this time they were downregulated here in the caruncule. And here in the cotyledon, we also thought it was interesting to see these processes related to arachidonic acid metabolism being upregulated. And among those genes, we found the prostaglandin E synthase here being upregulated too. So this agrees with previous findings. But when looking to the downregulated processes, we did find the prostaglandin receptors being downregulated, and between these genes, we found the prostaglandin E receptor being downregulated too. So still talking about the downregulated processes, we had genes related to post-embryonic development, placenta development, again, hyposignaling and MAPK signaling related to early embryonic development, and also a pathway related to angiogenesis here. And in the caruncule, besides the genes related to extracellular matrix remodeling, we also had genes related to placenta development being downregulated. And about the PEGs, they again were not differently expressed between the groups, but they were being lower expressed in the low PEG group. So with these findings, we conclude that the signs of potential late embryonic mortality are not evident on the trophectoderm on day 25, but we could see poor immune and vasculogenesis processes enriched in the caruncule. When we go to day 36, we can see that the low PEG environments are influencing the reproductive transcriptome, as we could see by processes related to poor embryonic development, poor placentation, increased apoptosis processes, low regulation of vasculogenesis and angiogenesis, and also differences in the prostaglandin E profiles. Higher slow PEG environments did not seem to affect changes on the CL transcriptome on days 25 and 36. So I would like to thank all the students and professors that collaborated with this project, and thank you for the opportunity to presenting here today, and I will take any questions. Thank you. So the PEG functions are not very well established right now. We do have some hypotheses that PEGs could be related to tissue remodeling, which we found those differences between high and low PEG, and also immunomodulation. And we did find some immune responses being downregulated in the low PEG group. So I believe that they're being caused by the PEG environment. Other hypothesis that we had about PEGs is that they could be luteotropic. So that's why we were looking to the CL2. But then we saw progesterone concentrations were not different and very few differently expressed genes. So I think we still have to keep investigating the CL. We did the same experiment on day 40 in Bostaro's cows, and we had more DEGs on the CL, like we had maybe 100 DEGs. So we're still looking in the CL, but we have a long way to go. It could answer that question. And other study that we're planning is to actually remove PEGs from these embryos and transfer these embryos and see how the pregnancy will follow. That's a very good question, Dr. Perry. We're still investigating that. I thought it was very interesting because on day 25, we couldn't see anything related to that intertrophectoderm. And if there's something wrong with early embryonic development, we would probably see something sooner. So yeah, we're still trying to investigate that and see where would they be interfering in the embryonic development and what would this cause. All right, thank you.
Okay. For this next time slot, we have a tag team going to present. We have two, let's see, the residents in embryo in the equine Physiology. Okay. First talk is by Camilo Hernandez Aviles. He's going to talk about studies in acrosome function, fertile and subfertile stallion. Sir, thank you. Actually, need to look at my presentation. Sorry about this.
There you are. Well, good morning, all. Thank you for everybody for having us here. Thank you, Dr. Johnson, for extending the invitation to Dr. Love. So today I'm going to talk about studies in the acrosomal function in fertile stallions, which is part of my PhD dissertation. To start, in the early 2000s, our group described the occurrence of a condition that affected certain stallions, particularly young stallions, which had very reduced sperm fertility rates, but in which the conventional sperm quality parameters were not able to detect or to explain why the stallions were dramatically fertile. These stallions were identified that have an acrosomal dysfunction, particularly because their sperm was treated with calcium ionophore 823-187, which is a molecule that is widely studied to induce acrosomal exocytosis, and most of these stallions were not able to acrosome react after incubation with this chemical. Also, our group found out that sperm from these stallions had a higher cholesterol to phospholipid ratio in their membranes, which kind of like makes sense and might explain why their acrosome did not react as fast and steadily as sperm from fertile stallions. In 2012, we took samples from these 7 initially identified stallions, and we conducted a genome-wide association study that identified a susceptibility genotype related to this condition. And I want to make emphasis that it's a susceptibility genotype, not a causative reason of this acrosomal failure. So every time that we got a stallion that has these reduced fertility rates and which we identified that they might have reduced acrosomal exocytosis, we genotyped their hair samples or blood samples, and we found out that for the gene FKBP6, which is located in the chromosome 13 of the horse, there is a double SNP in the exon 5. In this case, is the SNP. Oops, sorry. What happened? There we go. Sorry. And we found out that in this case, these stallions, every time that they have the susceptibility genotype AAAA, they were subfertile. So we wanted to figure out how frequent was this condition in the population of Brigham stallions that we try to evaluate every single year at Texas A&M University. In this case, we conducted an analysis on 1,100 different breeding soundness examinations or semen samples that were analyzed in our laboratory, in which the complaint was that the stallion has reduced fertility as per the stallion owner or agent or the referring veterinarian. And in these stallions, we conducted conventional semen analysis, namely sperm motility analysis, morphology, sperm DNA integrity, and sperm viability. From all these 1,100 stallions, we only found 21 stallions that had reduced fertility. Reduced fertility is a cutoff value of 50% or less of per cycle pregnancy rates, which corresponded to about 1.8 to 2% of the entire population that we analyzed. And in these 21 stallions, we were not able to identify any abnormality that explained the reduced fertility rates. For these 21 stallions, we did our calcium ionophore induction of acrosomal exocytosis, but instead of using transmission electron microscopy, which was a technique that was developed back in the early 2000s, we used flow cytometry. And we found out from these 21 stallions, only 8 had reduced acrosomal exocytosis rate. So from these 8 stallions, we found that 5 had what we call a questionable acrosomal exocytosis rate, so we're kind of like borderline, and only 3 had dramatic reduced acrosomal exocytosis. Look this in a different way. So a stallion that has an acrosomal response that we call acceptable is the one in which more than 70% of our sperm acrosome react after being treated with calcium ionophore. A questionable is between 50 and 69%, and abnormal response is less than 49% of the sperm that acrosome react after being treated with calcium ionophore. We also took samples from these stallions and we subjected them to Sanger sequencing to determine the genotype for the gene FKBP6. And we found out that from the 8 stallions, 7 of them were thoroughbreds and they had the susceptibility genotype for acrosomal reaction failure, while just 1 of them, which was a Frisian, did not have this genotype. And we found out that even though the calcium ionophore is a well-known molecule to induce acrosome reaction in stallion sperm, it's not a physiological method. So we noticed that there was a sperm death during incubation, which makes it a little bit challenging if we want to study further this condition. This graph shows that on the right panel, so we notice differences in the acrosomal reaction in sperm from fertile and subfertile stallions, but in both stallion groups, we found out that the sperm were dying even after 1 hour of incubation with this molecule. So obviously we needed to find out a better way to try to study acrosomal function in sperm from these animals. And that's how we tried to validate a more physiological method to study acrosomal exocytosis. And this came from a question that my colleague, Dr. Ramirez, tried to solve when she was working with Dr. Hendricks to find out which type of energy substrates may or may not modify the acrosomal response of sperm incubated under capacitating conditions. And we found out that having the sperm incubated in a medium with just lactate as energy substrate is still in the sperm without having to use neither progesterone nor calcium ionophore, and that this medium did not affect the sperm viability over to 4 to 6 hours of incubation, which is kind of like very physiological to us. So we tried to compare this lactate model versus the widely known calcium ionophore method, and we found out that, again, the viability of the sperm after being incubated with the calcium ionophore was dramatically reduced using the calcium ionophore treatment after 2, 4, and 6 hours of incubation, but this lactate medium did not affect the viability of the sperm incubated with these molecules. On the other hand, and more interestingly, only the lactate medium had a dramatic increase on the percent of sperm that were both viable and acrosome reacted after 6 hours of incubation as compared with control treatment or the calcium ionophore treatment. Then we wanted to investigate whether or not we can use these type of models with different types of semen, considering that in the equine industry, most of the mares are bred with cool semen or frozen thawed semen. And having frozen thawed semen for conducting this type of studies in subfertile stallions is more convenient for us. Instead of having the stallion sent to us, we can just have their frozen semen. And we found out that, here, we found out that cooling the semen or freezing and thawing did not negatively affect the capacity of the sperm to respond to this stimuli, which was really handy for us. We also tried to relate the responses of the sperm to this model to the fertility rates of the stallions that were used in the program with artificial insemination with cool semen. And while we were not able to detect numerical differences in the response of the sperm to the calcium ionophore treatment, we were able to detect numerical differences when we used the lactate model, and these differences were related to the pregnancy rates of the stallions. So stallions with the higher fertility rates were also the ones that had the higher response to our model. So finally, we wanted to identify whether or not we can use this lactate model to detect the responses of sperm from fertile and subfertile stallions that we knew that had this susceptibility genotype, and we found out that obviously the stallions that were subfertile had a dramatic lower value of acrosomal exocytosis in viable sperm as compared with the fertile stallions. Based on this, we decided to move to the next step, which was studying the sperm proteome in the subfertile stallions using mass spectrometry. So we used frozen semen from 3 fertile thoroughbred stallions that were located in Kentucky, which we had access to, and we had records of fertility, and 3 subfertile stallions, which were also in Kentucky, knowing that we were able to the sperm from both stallion groups under our defined capacitation conditions, and we use flow cytometry from the fertile stallion was not acrosome reacting, and we also used data independent acquisition mass spectrometry by what were the differences in the proteome between approaches. We identified 28 differentially abundant proteins compared to the fertile stallions, and these 20 identify up to 96% of the variation that of these 28 differentially abundant fertile stallions as compared to the fertile ones, while just with the fertile stallions. In the analysis, we found out in the subfertile stallions were related to leak, and based on the genome, the 2 lysosomal components, acrosomes, lysosomal. For example, here I just brought 2 proteins that were are related to lipid metabolism. On the left panel in the fertile stallions, and on the right panel for the fertile stallions, were acrosome reacting in the based on all these studies, acrosomal exocytosis failed to this FKBP6 very low frequency in the stallion bred horses. Method to start to study clinical and we have identified at the beginning membranes of the sperm from metabolism. The candidate proteins, which is where the little link between the with the have worked with us. Questions or no, Dr. It's interesting that you mentioned that because know that some of the fertile but there are relationship between soil removal from a with these genotypic cause. But thank you.
Has been increased the plus B research and where it due to that like
for example. They freeze and as the improve species cat study the focus and be the to the take information and we um, with on to the last we are picking we
We talking about how English teach in the year and there. Of it. Area. The years. We have where it's and that
way. Increases of culture rate and lactate aerobic life. One carb the glycerate since it's passive in light can be late by we are time some from the you can convert in our level the maybe and only I
And the glue trans expression to express and then an input are two sides. The
time. Listen. Just, over and how in of these social and epithelial cells starting as fully the best method. And also the use of the seminiferous is to this variations. So the exactly, however, did the rest. Do this do it and so we time uh, say value with the high micro million like this is what the not interest we can um access or photo can to the we we we've seen uh architect- integral have to the uh, we've with yeah. Soon we please with as with the traffic is not have